Okay, so welcome back to this next video in which we are discussing the digestion and absorption of fats. Okay, so we're currently in the process of discussing the components of bile, and we want to discuss what bile acids are. So basically, they're all sort of similar structures. They're all steroids, and most of them are based on the structure of cholic acid. Okay, so I want to give you an example of a bile acid which is based on the structure of cholic acid, which is taurocholic acid. Okay, and basically taurocholic acid is what you get when you combine cholic acid, shown here, with taurine. Okay, so let's firstly talk about the structure of taurine. Okay, so basically, taurine is the old name for a molecule which would now be called 2-aminoethane sulfonic acid. Okay, so 2-aminoethane and then sulfonic acid. Right. So basically, although this might sound complicated, it only sounds complicated because of this sulfonic acid. What does that mean? So let me show you what that means. So we'll start with ethane, so that's just a 2-carbon hydrocarbon, okay, like so. And then off one of the carbons we want an amino group, so off the one that we will call the second carbon, we want an amino group, like so. And then off the first carbon we want a sulfonic acid group. Okay, so basically the structure of a sulfonic acid group is like so. You have a sulfur atom with 2 oxygens that are both double bound to it, and then an alcohol group here. And basically the reason that this is considered an acid is that this alcohol group here can donate a proton away. So again, it's very similar to a carboxylic acid group. If you just look at this portion here, so if I get my highlighters again, if you just look at this portion, that's very similar to a carboxylic acid group. So indeed, the proton on this alcohol group can be donated away, leaving this oxygen with a negative charge, and therefore the molecule that I have shown here is indeed an acid. Okay, right, so what we're going to do is we're going to bind this taurine molecule, or 2-aminoethane sulfonic acid, to our cholic acid molecule via an amide link. So you might think, are we going to use the sulfonic acid group? No, we're not. We're going to use this carboxylic acid group of the cholic acid, and we're going to bind it to the amino group of our taurine molecule. So basically we're going to form what's called an amide link. Okay, we're going to take the alcohol group off that carboxylic acid group up here. We'll take the hydrogen off the amino group here, combine the two together to make water, and then bind the carboxylic acid carbon up there to the nitrogen here. So let's now draw the structure then of taurocholic acid. So I want to kind of try and keep both of their structures in view. Okay, so let's draw taurocholic acid first. So we'll start off with the basic steroid structure. So we've got these 4 carbon rings here. So we've got 3 6-membered carbon rings. Okay, so here's the third, and then this 1 5-membered carbon ring here. Okay, right, and then we want a methyl group off this carbon, a methyl group off this carbon, alcohol groups coming off 3 of these carbons, 1, 2, and then the sterile alcohol group down here. And then we have our side group off here, which is this 5-carbon carboxylic acid group. Okay, and we're now going to go over to drawing skeletal structures for carboxylic acid groups. So here's the carbonyl group shown skeletally. It will then be bound to the nitrogen of the taurine molecule, which will still have a single hydrogen attached. We're now drawing skeletally, so this first carbon here will just be a corner. Here's the second carbon here, and now we draw our sulfonic acid group here. So we'll have a double bond to an oxygen, another double bond to an oxygen, and then finally we'll end with an alcohol group. So this is the structure of taurocholic acid, which is an example of a bile acid. So this is one of the things which is contained within bile, basically. Okay, so it's an example of a bile acid. Right. Okay, so we're not going to see the function of the bile acids yet. They will come when we're actually looking at absorption. We haven't actually done digestion yet, so let's now talk about digestion. So we've done stage 1, which is this emulsification. We have secreted the bile into the duodenum. That has been mixed in with the chyme that we've received from the stomach, and we have had this huge injection of lecithin, which will help split the large fat globule down into many much smaller droplets, basically. Okay, and that will increase the surface area of the fat by around 1,000 times, which means that our enzymes now are going to have a much better shot at actually being able to digest some lipid molecules, basically. Okay, so we now want to talk about the different enzymes which are going to be involved in the digestion of the different types of fat molecules. Okay, and all of these enzymes which are going to be involved in the digestion of the lipid molecules, they're going to come from the exocrine pancreas. Okay, where's my picture of the pancreas gone? So here is the pancreas. So remember, the pancreas has both an endocrine function and an exocrine function. Most of the cells within the pancreas are devoted to producing digestive enzymes which will be secreted into the pancreatic ducts, which will then secrete those into the lumen of the duodenum, basically. Okay, but there are other clusters of cells within the pancreas, known as the islets of Langerhans, which have an endocrine function, which we're not going to discuss here. So basically, the exocrine cells of the pancreas will be secreting digestive enzymes, which will be making their way through much smaller little tubes into these large ducts that I've shown here, which will then go into the intestine. Now, they will be secreted initially as proenzymes, so that they only become activated once they actually get into the duodenum. So, basically, let's start with our discussion of the enzymes which will digest the different fat molecules that we've discussed. So we'll start with the most important one, which is the triacylglyceride molecules. Okay, or rather, sorry, triacylglycerol molecules, triglyceride molecules. Okay, right. So here we have our 3 long-chain carboxylic acid molecules attached to our glycerol molecule. So basically, we have an enzyme which can break down triacylglycerol molecules, and this is called pancreatic lipase. Okay, so this will be secreted by the pancreas. Now, what's it going to do? Well, basically, it's going to hydrolyze the ester links between the long-chain carboxylic acids and the alcohol groups of the glycerol molecule. Okay, so what is this going to result in the production of? Well, basically, pancreatic lipase can break off 2 of the long-chain carboxylic acids. Okay, so it'll start with this one up here. So, basically, what it will do is it will turn the triacylglycerol here into what's known as a diacylglycerol. So when you just have 2 acyl groups attached to the glycerol molecule, that's called a diacylglycerol, often abbreviated to DAG. So this is short for diacyl and then glycerol. Okay. So we go from a triacylglycerol, a TAG, to a diacylglycerol, a DAG, and we also produce a free fatty acid, as called. So when you have long-chain carboxylic acids, which are not attached to anything and are just floating around free, these are called free fatty acids. And free fatty acids are often abbreviated to FFAs for short. So the first F is for free, the second F is for fatty, and the A is for acid. Okay, right. Now, pancreatic lipase isn't going to stop there. It can break another one of these off. And basically, it also breaks off the other one that's on the other side. Now, if you think about this, this makes sense. Okay? Pancreatic lipase has a certain active site. Okay, now it has broken off one of the long-chain carboxylic acids that is on the end of a glycerol molecule. Okay, so which of the two would you expect it to be able to break off now? Well, how is it going to be able to break off this one here? Because, you know, that's not on the end of the glycerol molecule, that's suddenly in the middle, okay? So it's far more likely that it will be able to break off this other one that's over here than the one in the middle, because this one looks just like that one, basically. Okay, so indeed it's going to break off the other one that's to one side, basically. Okay, so what you'll now get is this here. So we've got our long-chain carboxylic acid in orange here, and then our glycerol molecule in green, and now we'll get another free fatty acid molecule here in orange. Okay, so the second reaction is also catalyzed by the pancreatic lipase enzyme, so it catalyzes both this first reaction, where we go from the triacyl glycerol to the diacyl glycerol and free fatty acid, and then it's going to take the diacyl glycerol and it's going to convert it to another free fatty acid and this third molecule here, which is basically called a monoacyl glycerol. Okay, and monoacyl glycerol will often be abbreviated to MAG. Now, some people will call this 2-monoacyl glycerol to denote that the single acyl group that you have is coming off the second alcohol group of the glycerol molecule. So you might see this referred to as 2-MAG, just to be rigorous. Okay, right. Now, pancreatic lipase can't go any further. It can't break down 2-monoacyl glycerol into glycerol and a final free fatty acid molecule, but it doesn't need to. The 2-monoacyl glycerol can be absorbed as it is, so you don't need to break it down any further. So, basically, we've secreted this enzyme into the lumen of the intestine, and it's going to work on the triacyl glycerols within the lumen of the intestine, and it's going to convert them overall into 2-monoacyl glycerol, and then we'll also have 2 free fatty acid molecules, one here and one over here. Okay, and these are then going to be absorbed, and we'll discuss the absorption in a moment. Okay, right, let's now discuss the digestion of the phosphoglycerolipids. Okay, so let's draw our phosphoglycerolipid like so, and this will include, by the way, all of that lecithin that we secreted from the bile. We're not just going to, waste it and let it pass out of the body. We're going to reabsorb it and then reuse it, basically. Let's draw our phosphoglycerolipid here. Okay, so here are 2 long-chain carboxylic acids shown here as these horizontal orange lines. Then we have our glycerol molecule shown here as this vertical green line. We have our phosphate group shown here as this pink blob. And we then have the additional structure that's attached onto the phosphate group of the phosphatidic acid molecule. Okay, right. So, this, for example, could be choline, and indeed a lot of it will be choline, because we've secreted a huge amount of phosphatidyl choline into the intestinal lumen. Okay, so this is a phosphoglycerolipid. Okay, right. So basically, you secrete an enzyme which is a phospholipase A2 enzyme into the lumen of the intestine. Okay, so this will be a phospholipase A2 enzyme, and for short, phospholipase A2 enzymes are abbreviated to PLA2, like so. So P for phospho, L for lipase, and then A2. Now, what do phospholipase A2 enzymes do to the phosphoglycerolipid? Well, basically, they're going to hydrolyze the ester link between the long-chain carboxylic acid that's in position 2 and the alcohol group of the second carbon of the glycerol molecule. And what they'll overall create will look like this. Okay, so you'll have one long-chain carboxylic acid still bound. You'll then have your glycerol molecule, your phosphate group, and whatever additional group has been added on, whether that's choline or ethanol-amine or glycerol or inositol 4,5 bisphosphate, whatever it may be. Okay, so let's colour this in. So here's our glycerol molecule, here's our long-chain carboxylic acid, and here's our phosphate group of the third carbon of the glycerol molecule. Okay, and of course the other product of this breakdown is that you also get a free fatty acid molecule. Okay, right, so what is the name for this other molecule that we've created here? Well, basically, you'll have a different name depending on the exact phosphoglycerolipid you are talking about. Okay, so for instance, if we were talking about phosphatidylcholine and this blue molecule therefore was a choline molecule, then this would be called lysophosphatidylcholine. So the P is for phosphatidyl, the C is for choline. If we were talking about phosphatidylethanol-amine, then we'd call this lysophosphatidylethanolamine. If this was a glycerol molecule instead of an ethanol-amine molecule, then this would be phosphatidylglycerol. Then this molecule here would be a lysophosphatidylglycerol molecule. Okay, right, so that's the digestion of the phosphoglycerolipids, and finally we want to discuss the digestion of the cholesterol esters. So basically, the cholesterol molecules which are free and don't have a long-chain carboxylic acid molecule attached to them, they are fine, they can be absorbed directly as they are, but the cholesterol esters need to firstly have that ester link hydrolyzed, basically. Okay, so let's just draw out the structure of our cholesterol ester again. So let's start with our steroid structure here. So here are our 4 carbon rings. So here is our second 6-membered carbon ring, our third 6-membered carbon ring, and then our 5-membered carbon ring up here. Remember, in cholesterol we have a double bond there, an alcohol group off here, a methyl group there, a methyl group there, and then we have this 7-membered carbon structure up here. Okay, so this is cholesterol. We want cholesterol esters, however, so let me just take that hydrogen off here, and we'll attach on some long-chain carboxylic acid here. Okay, so basically what we're going to do is hydrolyze that bond to yield cholesterol. Okay, and I won't draw that out again. So cholesterol, and then just a long-chain carboxylic acid, which I'll draw in my cartoon way, just as a line that I'll colour in orange, like so. So, what is the name of the enzyme that's going to carry out this conversion, this hydrolysis reaction? Well, it's basically an enzyme called cholesterol ester hydrolase. Okay, so this is cholesterol ester hydrolase, which is a name that makes full sense because it's going to hydrolyze cholesterol esters. Okay, so over here, this structure that we have here is a cholesterol ester. And of course, again, there's not just one molecule which will be called a cholesterol ester, because this R group that I have here is effectively arbitrary. That can be any old long-chain carboxylic acid. So there's a great many different cholesterol esters. Okay, so cholesterol ester hydrolase will then break down the cholesterol esters into cholesterol and long-chain carboxylic acids. Okay, right. So, now what we have had happen is we've had the large fat droplet being emulsified down to loads of little tiny droplets. Then these free enzymes, pancreatic lipase, phospholipase A2 from the pancreas, and also the cholesterol ester hydrolase from the pancreas, they've all gone to work basically, and they've started breaking down the triacyl glycerol molecules into 2 monoacyl glycerol molecules. and 2 free fatty acid molecules, and they've broken down the phosphoglycerolipids into phosphoglycerolipids, although that's not common terminology, and free fatty acids. And finally, the cholesterol ester hydrolase has broken down cholesterol esters into cholesterol and free fatty acid molecules. So this is a free fatty acid molecule. Okay, we are now going to take the 2-monoacyl glycerol, the free fatty acids, the lysophospholipase, and the cholesterol molecules, and we're going to absorb them. Okay, now this is where the bile acids are going to become important. So let's now discuss how the bile acids, which we saw an example of here, taurocolic acid, are going to actually be important. So basically, there is a problem, okay? These enzymes, for instance, pancreatic lipase, phospholipase A2, and also cholesterol ester hydrolase, they are reversible, okay? Well, the reactions they're catalyzing are reversible. They can catalyze them in both directions. So, if we want to actually get all of our triacyl glycerols, for instance, broken down into free fatty acid molecules and 2 monoacyl glycerol molecules, we basically are going to have to remove the breakdown products before they can be reconverted back into the original molecule that we can't absorb. So basically what we need to be doing is collecting the breakdown products, okay, from these digestion reactions and getting them to be absorbed quickly, basically. Okay, and basically the bile acids are going to help with the absorption of these molecules, and they're also going to help sequester these molecules away from the damaging enzymes which could take them and reverse them back into the original molecules. Okay, so basically the bile acids are going to be involved in taking these digestion products and taking them away from the enzymes which could turn them back into the original molecule. Okay, so basically what the bile acids do is they form structures known as micelles, okay, sometimes called mixed micelles. Okay, so they're going to form little structures called mixed micelles. So let me discuss with you what a mixed micelle is. So basically, a mixed micelle is a little sort of droplet, okay, which has a monolayer of these bile acids. Okay, so you're going to have a bile acid here, you're going to have a bile acid here, you're going to have a bile acid here, and it will go all the way around, basically. Okay, you'll have this monolayer, and it will make a sphere overall. Okay, now, I showed you the structure of these bile acids. So, for instance, I showed you an example of a bile acid. I showed you this taurocolic acid. You can see this sulfonic acid group up here. This could donate away this proton, and what would happen then? It would end up with a negative charge, i.e. it would become extremely polar. Okay, so the idea is that these bile acids can donate away their protons, because that's why they're called bile acids. They're all acids, okay, so they can donate away their protons, get negative charges, and they can basically position themselves so that the negative charged group is sticking out towards the outside of this micelle, whilst the rest of the hydrophobic molecule is facing into the middle. Okay, so that's the basic idea of these mixed micelles, that the bile acids have these polar groups which can face the outside, okay, and then their hydrophobic portions can face the inside. So, these things that I've drawn here, these are all bile acids, which I'm now colouring in orange. Okay, right, so basically, as soon as you put bile acids in water, they will form these micelle structures, okay? And what can now happen is all of our breakdown products, so for instance our free fatty acid molecules, which I've always shown in orange, and therefore I'm not going to change, even though I've just now shown all the bile acids in orange as well. Then what other breakdown products do we have? We have our 2 monoacyl glycerol molecules. Okay, so here is 2-mag. So I'll colour that one in as well. Here's that single long chain carboxylic acid, and here's our glycerol molecule. Okay, we then have our lysophosphoglycerolipid molecule here. So here's our glycerol molecule, here's our phosphate group, and then here's our extra additional head group, whatever it may be. And I should stress that it's not necessary that you have that head group. It could have originally just been a phosphatidic acid molecule, in which case what we would now have is a lysophosphatidic acid molecule. Okay, and here is the glycerol molecule. Here is our phosphate group coming off the 3rd carbon's alcohol group, and here is that additional group that you potentially have added onto the other alcohol group of that phosphate group of the phosphatidic acid molecule. Okay, so this is our lyso, and I will put phosphoglycerolipid, although that isn't common nomenclature. So lysophosphoglycerolipid, PGL for phosphoglycerolipid. Okay, so you know what I mean there. The breakdown product from our phosphoglycerolipid. Okay, right. And then finally we'll have the cholesterol molecules. Okay, so I'll just draw the little skeletal structure of cholesterol here. So here is our steroid structure with these 4 carbon rings here. So this is another 6-membered one. Here's our 5-membered one. Here's our methyl group. Here is this 7 carbon structure up here. And then we've got our double bond there and our alcohol group here. Okay, so all of these things are basically going to come in and join the mixed micelle, which at the moment just consists of the bile acids. Okay, so what's going to happen is basically the 3 fatty acids are going to go into the center here, the 2 monoacyl glycerols will go into the center here, the lysophosphoglycerolipids will sort of position themselves so that they're near the monolayer basically, so that their phosphate groups and their heads can stick out and join the polar groups of the bile acid molecules and also face onto the water that's on the outside here. So let's just put a water molecule out here. And then cholesterol, it can also sort of position itself in the monolayer, and basically it will have this alcohol group which is slightly polar, sticking out towards the outside to interact with water molecules. Okay, so that's the structure of these mixed micelles. So basically, overall what's happening now is the digestion products from our fat molecules are going into these mixed micelles, and what's now going to happen is these mixed micelles are going to take the breakdown products to the epithelial cells, to the enterocytes, and there what will happen is the digestion products will enter the enterocyte, and the story will continue from there. Okay, so we'll call it there for this video. And in the next video, what we'll look at is the histology of the stomach wall, so that we can then understand how absorption is going to happen. Because digestion is now finished, we now need to discuss absorption.